After talking about the recombinant DNA technology, let's move on to DNA fingerprinting. You may ask, how does DNA fingerprinting work? The base sequence on DNA is nearly the same in all humans, about 99.9%, while the remaining 0.1% contains some regions that are variable in length for different individuals. DNA fingerprinting is developed to identify individuals by analyzing the length of these regions. There are 4 major steps in DNA fingerprinting. First, extract DNA from collected samples. Blood, semen, and hairs are the common sources of DNA. Second, the DNA collected are mixed with enzymes that will cut the DNA into fragments. The fragments with various variable regions will be obtained. Third, by using electrophoresis, the DNA fragments are separated according to their sizes. Shorter fragments move faster towards the positive terminal than longer fragments. Finally, DNA fingerprinting, which is shown as a pattern of bands, is produced, which is unique to each individual. So, what is gel electrophoresis? It is a technique that uses electric fields and gel slab to separate DNA fragments. DNA fragments are negatively charged due to the phosphate groups present. Under an electric field, all DNA fragments move towards the positive terminal. There are many molecules spaced in the gel slab, which allows shorter DNA fragments to move faster and travel further than the longer ones in a fixed period of time. Eventually, DNA fragments are separated into bands according to their sizes. Staining with fluorescent dye is required before the DNA bands produced in the gel slab can be seen under ultraviolet light. We can copy the pattern of bands by photographic film for a longer lasting record. How can we apply DNA fingerprinting in our daily life? First of all, it provides evidence on the identities of individuals in court cases, like rapes and murders. As the DNA fingerprints of everyone are unique, the DNA sample collected at the crime scene can be strong evidence for crime. Second, it establishes family relationships in parentage tests. It is based on the fact that half of the genetic materials of an individual comes from the father and the other half from the mother. Therefore, each individual shares half of the bands in his DNA fingerprint with the father and half with the mother. Third, it can identify victims of disasters. By comparing the DNA fingerprint of the dead body with that of the suspected victim, victim identification can be done after earthquakes or tsunamis. Next, it can provide authentication of Chinese medicines. Substituted Chinese medicines are commonly found in the market that will harm our health. In order to ensure public safety, DNA fingerprinting can be used for authentication by comparing to genuine medicine. Finally, an example is conservation of endangered species. Some animals have become endangered due to overhunting, like African elephants are often killed for their tusks. DNA fingerprinting is a useful tool to identify the origin of IV products. It helps authorities finding out which country elephants have been killed and aids them in arresting the hunters. Biotechnology not only boosts the development of DNA fingerprinting, but also Human Genome Project. What is the Human Genome Project? A genome refers to the complete set of DNA of an organism, including all its genes. Understanding all the genes present in humans would be of great benefit, as genes determine our body characteristics. Therefore, the Human Genome Project is mainly to investigate the genes in the human body with the following goals. First, determine the base sequence in human genome. Second, for human genome mapping. Third, for DNA information storage. And finally, related new technologies development. It was first started in the US in 1990s. Then it involved scientists from more than 18 countries. Here, we will show a brief timeline of some achievements of this project. In the first year, 1990, the first gene for breast cancer was mapped. In 1996, sequencing of yeast genome and mouse genome was completed. 3 years later, in 1999, sequencing of the first human chromosome was completed too. In the year 2000, the draft version of human genome sequence was completed, and also genome of fruit fly. In 2003, the complete version of the human genome sequence was finished, and other analysis of data continued in the following years. All the goals of the project were achieved 2 years earlier than expected. Meanwhile, many other international genomics projects are still undergoing, like the Cancer Genome Project, which aims to identify the genes critical to the development of cancers in the human genome. Data from the Human Genome Project may bring us many benefits. The information from the project can help scientists better understand how genes control biological processes in the body, how genes are inherited and how genes mutate, and also find out the functions of specific genes. Second, new treatment methods and diagnostic tests can be developed based on the gene data, such as breast cancer and Alzheimer's disease. Third, we can have a better understanding of evolution by comparing the genomes of different organisms and can help build up their phylogenetic relationships. However, there are some limitations to the data obtained from the Human Genome Project. Although the project had sequenced and located many genes, the functions of many genes are still unknown. Moreover, the data obtained from the project cannot explain the brain function and some biological processes. On the other hand, it has raised ethical, legal, and social issues. Should employers have the right to access employees' genetic information? Would you like to know if you have the gene for an incurable disease? Relationship of biotechnology with the Chinese Mitten Crabs aquaponics project. The Human Genome Project has inspired other genome projects related to other different species. In 2017, Chinese scientists published a draft genome of the Chinese mitten crab, Erosia sinensis. When looking at the genome, the scientists discovered that there are a total of 1.12 gigabase pairs. Its genome is about a third of that of human, which is 3.1 gigabase pairs. The group of scientists identified 14,436 genes. This first genome sequence of the Chinese mitten crab provides a valuable resource for the study of essential developmental processes and genetic determination of important traits of the Chinese mitten crab, and also for the investigation of crustacean evolution. In a further study on crustacean evolution based on Chinese mitten crab's genome project, genetic analysis comparing Chinese mitten crabs and king prawn Panacea viname showed that Chinese mitten crabs evolved separately 373.6 million years ago and at a much higher evolutionary rate than king prawn. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. We'll see you next time. Bye bye.